Insertions and deletions are called frame shift mutations because they shift the reading frame of the genetic message. Reading frames are 3 base pairs long. In this example, we have a deletion. In the mutant sequence, we are missing a U. This changed the amino acid from phenylalanine to leucine. In phenylalanine, we would read UUU, but because we are missing a U here, we read UUG. This also shifts the next amino acid. Instead of reading GGC that codes for glycine, we will read GCU that codes for alanine. We have an insertion of cytosine. So in the mutant DNA, we can see that it reads CCT instead of CTT. Once we transform these sequences to line up with our codons, which will have uracil instead of thymine, we will see that this changes our last 4 reading frames, which code for different amino acids.
Amino acid, for example, this specific sequence of amino acids resonates with manufacturing factors.
Right there!
Let's walk through an example together. In deletion, we will start with the normal and mutant sequences. We will then look to our Codon Dictionary and place the appropriate codon sequences underneath each amino acid sequence. For methionine, the codon associated with it is AUG. We will place our AUG here and our AUG here as well. Next is the amino acid TRP. TRP goes with UGG. We will place UGG here and UGG here. In the normal sequence, the next amino acid we have is phenylalanine, but we have isoleucine in our mutant sequence. This is where our mutation took place. What I like to do is finish out the normal sequence, to the same to our mutant. So let's see if it's a deletion. We can try to delete the U, but that will give us UC. Isoleucine has an A in front of these sequences, so a deletion would not work. So let's try an insertion of an A. If we insert an A into these sequences, we could get AUU. Does isoleucine have an AUU? It sure does. So we will be using AUU as part of the new sequence. Since we used AUU for our mutant sequence, the reading frame of the mutant sequence would stop here. Next, we could either get a CAC or a UAC. Histidine has a CAC, so we will be using this in the sequence for the mutant sequence. We must add our stop codons. Looking at our Codon Dictionary, here are our stop codons. We will place them underneath and see which stop codon the mutant sequence will have. Since we stopped, and our reading frame stopped here at CAC, we could either have a U, a C, a or a G to begin the stop codons. But as you can see, the stop codons start with the U. So we must be using this U here, and we're going to read our 3 base pairs, and we can see that the stop codon that we'll be using for the mutant is UAA. Sequence for the mutant codon, including the stop codon UAA. For the normal sequence, we do have to go off the mutant sequence. So we used AUG, we use UGG, and for this one we use UCC because AUU is used in the mutant. Now the next one in the mutant is CAC, which means that we must have used ACU for the normal sequence. The next one in the mutant is UAA, so that means we could either use AAA or AAG because the reading frame stopped with these 2 AAs. You can pick either one. Since the reading frame stopped at AA, you can use any of the codons listed for the rest of the amino acids. In my sequence, I used AAA and GGU.
How you get that ball?